Hi everybody, and welcome to my demo for running DeepCell and QuPath together. This automates a really tedious first step of bioinformatics for cancer research. So just first of all, why are we here? What is all this stuff? Basically, what spatial omics lets us do is take microscope images of cell tissue, but then on top of that, understand what DNA is present, where the cells are, what kind of cells are there. It's a key frontier in medical research. The problem is that existing tooling is pretty hard to deploy, it's hard to use, and it's hard to integrate into existing pipelines, especially for the target user who's a bioinformatician or a scientist, not necessarily a DevOps expert. So this data goes through really complicated and long pipelines. Here's one example. Our little piece of it is actually just right over here. This is a process called SPAflow. The link is up here if you're curious. But again, our part is just over here on the left. We're going to identify, quantify, and measure the cells in some tissue that was imaged under a microscope. So that's what cellular segmentation is. So this part had already been launched before the build process. So we took this and we made it run on Google Batch. This outputs a NumPy array, which is visualized something like so. So you see here the image on the left now has outlines of cells on it. So this is the DeepCell process to use TensorFlow AI to identify the cells on these microscope images. Now, this is actually part of a much larger process called a QuPath project. And so that looks something a little bit more like this. We start with those microscope images, we turn them into the NumPy arrays, we detect the cells, and now that we have the cell boundaries, we actually measure them and do some analysis using QuPath libraries. And together this produces a project that's then ready for a scientist to analyze. So if we were to put it all together, our pipeline looks something like this. We took the DeepCell segmentation that we already had, and what we're launching here is the analysis step of the pipeline. So starting from our input images, those are TIFF files, going to NumPy arrays, going through cellular segmentation, getting those predictions of where the cells are, putting that through the QuPath analysis over here, step 2, to lastly end up step 3 with a QuPath project. So let's take this and actually see how it works. So over here, first of all, we would run the job by having this little helper script. It produces a bunch of JSON. You can see it up here. It's not super exciting, but that runs a batch job. Now, the batch job is not super exciting to watch, but here is an example. Once we have all this done, our starting point was here, the OMI TIFF directory. Those are the TIFF images there. We had the NumPy files here. And once this process completes, such as over here, we have this new directory called segmask. That's segmentation mask. And this contains those predicted TIFFs. If we come over here, here we are at a local copy of the input files. And if I were to list the contents of where we were just looking, that's what we get. So let's copy this here. Let's copy that segmask directory here. Now we have a directory that contains the OMI TIFFs, the segmasks, and the intermediate. These input files, just to show you what they look like, here we are in OMI TIFF. Let's take number 3, for example. They're imaging, those look like cell nuclei, and then this is cell membranes, which are the borders between tissue. So that's what these look like. Then we come over here, and we are now in the analysis part, the part that runs the QuPath code. And I'm just going to go ahead and run this Java code. You can see it doing the analysis. This part doesn't yet run in the cloud. The other part runs on batch. This part we're currently working on containerizing. Anyhow, now over here we have QuPath. So here's the QuPath application ready to go. I'm going to open the project and I'm going to select it right there. Open. And now we have those TIFF files that we were looking at previously, but this time if we open them up, they actually have the annotations loaded up into QuPath. And so here you can do interesting things. I don't totally understand the science of it, but for instance, clicking on this cell, it's telling us that the mean intensity is 0.2. That's a relative intensity measure. It can tell us that is 3,000 square micrometers. And so from here, the scientists do their work. This is where I pass the baton off to the folks who actually know what they're doing in the research. But what I've hopefully provided is the ability to get started really, really quickly. So that's the launch. I'm going to come back here to the slides. This is the launch. It's going from the image files through Google Batch to generate segmentations, running the QuPath analysis, and lastly ending up with the project. Thank you so much for watching. I hope you enjoyed this.